I am a professor at the SPCI in Paris, and my laboratory study memory, and that's the reason of the title of this presentation. And while I was preparing this, I tried to focus way much more on the techniques and not on what we actually discovered using those techniques. This is like the core techniques that we are using in our laboratory. So it's very, and they are very cool techniques. So I hope you will get as excited, at least a bit excited as I am about those techniques. So right now I'm sharing the screen. I just want to check if you can actually see this, because I'm not able to see you.
Yes,
okay, perfect.
We can see.
Okay, so please, if you have any question, since I cannot see you, please just turn off your, unmute your microphone and go ahead with the question. So, so as the title said, what we are going to discuss today are two different methods, which is optogenetics and calcium imaging. And we are going to see also some examples of how you actually can use those techniques in neuroscience. So what is the main purpose of what we are doing as a neuroscientist, and in particular behavioral neuroscientist or cognitive neuroscientist, is to try to understand how the behavior of the animals or whatever of humans is correlating with the neuronal activity. And the relationship between behavior and neuronal activity is very tight. So obviously behavior is going to change the way that our neurons respond, our brain responds, but also our brain is obviously going to drive the behavior of us and of animal models. So trying to understand this relationship and how neuronal activity can change, can modulate the behavior and vice versa, is the main goal of behavioral neuroscientists in general. And so during this presentation, what I wanted to actually show you are two different techniques, which are like one of the most used techniques, but not the only ones, that allow us to both manipulate the neuronal activity and in real time visualize the behavioral response, and also visualize neuronal activity and correlate and be able to correlate it with the behavior of the animals. So during this presentation, during this class, I will show you two different methods. One that is manipulating neuronal activity, and the other one that is visualizing neuronal activity. And so I will start with the manipulation of the neuronal activity, introducing optogenetics. Probably several of you have heard about this technique, but maybe not all of you. So since the beginning of the journey of using optogenetics, which was actually a method that became method of the year in 2010, and it was published as a method of the year in Nature Methods, which is a very important journal. Since that moment, so in 2010, not long time ago, it really changed the way that we are studying the brain, and changed the way that we can actually visualize and understand behavior. So why is this the case? Why is such an important technique? Well, because thanks to optogenetics, it is now possible to analyze the real-time contribution of specific subpopulation of neurons, in particular behavioral tasks. So right now, we can actually modulate the neuronal activity just by using light. And this is the revolutionary aspect of optogenetics, is that we can turn on and off light. And while we turn on light, we can manipulate neurons. And when we turn off the light, we have no effect on the neuronal activation or inactivation. And we also, so we have like such a temporal resolution that was impossible to have before, but we can discuss also this one, because electrical stimulation is also very quick as light stimulation, but also the spatial resolution is another very important point about optogenetics, because we can target specific subpopulation of neurons and not all the neurons that are around the light. And I'm going to show you how this is possible. So what is the difference between, can you see my pointer moving? Can someone answer to me? No, you can't. Okay, so well, I'm gonna explain it to you even if you cannot see my pointer. The electrical stimulation, as you can see here in this graph in red, allow the possibility to activate neuron just by sticking an electrode in specific areas of the brain. So we can still manipulate those neurons, activate them. But the problem about electrical stimulation is that we cannot target specific subpopulation within that area. So everything that will be around this electrode will be activated. While using optogenetic, we can actually manipulate and modulate subpopulation of cells, we can manipulate it by exciting them or inhibiting them. And here you can see in this graph the excitation in blue or the inhibition in yellow, by using different receptors that we can engineerize and express in specific subpopulation of cells. So we can express those receptors, and those receptors are receptors that are light sensitive. So using light, we can then activate or inactivate those cells, depending on the type of receptor that we can express within the cells. So here there are like two representation of receptors, channelrhodopsin and then NPHR, which are the most common, actually the first ones, opsins that were originally used to manipulate neurons with light. And the channelrhodopsin is an excitatory opsin, so it allows the passage of sodium within its channel. And these channels are presented within the membrane of the cells, so that by presenting light, we can open this channel, and the opening of the channel will lead to the entrance of sodium within the cell. And with that, to the change of the potential of the cell in a very quick way. So just by turning on the light, and this turning on the light and opening the channel will allow the excitation of the cell. The cell will undergo through the action potential process and will be activated. And we can decide exactly just by turning on the light what is the time in which the cell will be activated. On the other side, we can have also different type of opsins, which are inhibitory opsins, which allow the entrance of negatively charged ions within the cell that will allow the possibility of inactivating those cells. And for these two examples, but there are several different type of opsins that are now available. We can choose between like a huge number of possibilities, depending on the type of experiment and on the type of precision and on the type of kinetics that we are looking for. We can actually use different lights for activating or for activating those channels, and indeed we can then excite or inhibit different cells depending on the type of opsin that we are using. So this is an example of microbes that are present in these opsins. So these opsins were first discovered, like the use of these opsins was first used from these microbials. So they are unicellular green algae. And through these photoreceptors, they can control phototaxis. So their movement, they are moving in response to light. So we can take those opsins from those organisms and we can engineerize them to be expressed in different organisms. Obviously, there are opsins, so photoreceptors present in different organisms, and they are necessary in these different organisms to control electrical excitability, intracellular acidity, calcium influx, and other cellular processes. So you can think about other type of opsins, and that are probably already in your mind. And those opsins are present again in different type of organisms, including, for example, bacteria, but also mammalians' brains. So, for example, in the cow, but even in humans, we do have in our retina rhodopsins, which are necessary for us to actually have the visual system working. So through light, these opsins allow the detection of light and the processing that allow us to have the vision. So there are different type of ion channels present into our brain or into different organisms' brain, into the neurons. And for example, some examples which you may already know are the voltage-gated ion channels, the ligand-gated ion channels, and again, the one that we are discussing today, which are the light-gated ion channels. So the voltage-gated ion channels, they open after a change in the membrane potential. The ligand-gated ion channels, they do open after the arrival of a neurotransmitter, for example. And the light-gated ion channels, they do open after stimulation by light. So as I said, these opsins, they do exist in our brain, well, in our eyes, but they don't exist in the rest of our brain, as for the majority of the organisms. And indeed, this is where the engineerized part arrives. So we do want to make some of the cells that we are interested in to become light, to be expressing these light-gated ion channels. There are also different type of ion channels that we can find into our system, and they are mainly part of our sensory tools. For example, we have chemoreceptors that are responding to chemicals. We have thermoreceptors that are responding to the temperature, mechanoreceptors as well, for example, the one that we have in our ears, and also the photoreceptors that, again, we do have into our eyes. There are, as I said, different type of opsins that we can use for optogenetics. For example, the channelrhodopsin is the one that is presented here as an excitatory opsin, but we do have like a bunch different type of opsins that are available right now, and also different type of inhibitory opsins. So as you can see here in the bottom, you have an example of a cell that is activated or inactivated just by using blue or yellow light. So this is just an example again, because there are some opsins that are excitatory opsins, but they are using the yellow light or the red light and not the blue light as in this example. But as you can see, if you look at the activity of the cell, you can see that by turning on the light, the blue light, this cell can be activated specifically only when the light goes on. And when you turn on the yellow light, you can see that there is an inhibition of the activity of the cells. So you can specifically control the activity of the cells just by turning on and off the light using those ion channels. So why those ion channels are important and why they can turn on and off the activity of the cell? Well, we should know a little bit about the action potential, which is the possibility, which is basically how everything that the neurons are doing is going through action potentials. And the reason why they use the action potential is to actually communicate with each other. Through communication with each other, neurons can actually, yes. Someone was asking questions? No?
I think just joined the meeting.
Okay,
so.
Cool. Okay. Okay, sorry, because I cannot see, so perfect. So what is an action potential is the way by whom neurons can communicate to each other. And the action potential reaches the end, the tip of the neuron, so the end of the neuron where neurons communicate to each other, so at the end of the axon, and they can reach the dendrite or the spines of a postsynaptic neuron. So what is happening during the action potential, and I don't want to go through all the details about it because it's going to be a completely different class, but eventually the first step of the beginning of an action potential is the influx, the entrance of calcium within the cells, and the possibility of releasing neurotransmitter between the pre and the postsynaptic neuron. And this leads to the activation of specific channels, which are at the, on the membrane of the postsynaptic cell, which will be opened and will lead to the entrance of positively or negatively charged ions, which allow the cell, the postsynaptic cell, indeed to change its the potential, the membrane potential, and indeed lead to the possibility of exciting or inhibiting the postsynaptic cell. So what are the ions that are important for this process, which are the ion channels that are present on the membrane? Well, to lead to an action potential, so to lead to excitability of a cell, there are two main ions that are important, which are the sodium and the potassium. And in particular, the sodium entrance within the cell is the one that is triggering the activation of the action potential, which is the reason why when we look at the channel that we discussed about optogenetic, which is the channel rhodopsin. The channel rhodopsin is particularly is an ion channel that allows the entrance of sodium. And so we can basically bypass, sorry, I'm going back here. We can basically bypass the normal activity of a cell, which allow the entrance of sodium within the cells through the activation of a neurotransmitter, just by engineerizing specific receptors to be expressed into the cell, which are not responding to the neurotransmitters, but they are responding to light. So how can we do this trick? How can we engineerize those cells? Well, we can, in general, we can use different techniques, but the main two techniques that are used are actually the expression of viruses within targeted cells, or the use of transgenic mice. So the expression of viruses is very convenient because it allows us to use normal animals and then inject viruses within the area of interest. And those viruses will be expressed, will be integrated in the DNA of the viruses will be integrated into the host cell that can then express the gene that we engineerized within the viral DNA. On the other side, we can also use transgenic mice, and in this case, we can also have different type of transgenic mice that are expressing our protein of interest. In this case, it's called the GCAMP, but it's just an example. In reality, what we are interested in is for optogenetics. We'll go back to the GCAMP, so this is also part of what I'm going to explain in the second part of the class. But in general, what we can do is to have transgenic mice that are expressing channel rhodopsin, for example, or inhibitory opsins under a specific promoter. So, for example, in this case, if you cross two different type of genetically transgenic mice, you can have the expression of your opsin under the dopamine receptor. And this means that you will have only channel rhodopsin or your opsin of interest under within the dopaminergic neurons. Here there is another example of a mouse, a brain, in which you can see the, an inhibitory opsin, which is the ENPHR, under the TIH promoter, which is a promoter that is present in all the cells. And so in this case, actually, you can have like opsins expressed within all the different areas of the brain. And then you can target whatever area of interest you want. So here is another example, and it's actually from one of the papers that we published in my laboratory, in which we were interested in a specific neurons, which are in the olfactory bulb, so in the olfactory system. And you can see how beautifully these specific neurons, and only those neurons, are present in the YFP, so the yellow fluorescent protein. And this is actually what you can visualize, but you cannot see. This is actually how these neurons are expressing. They are expressing not only the YFP, but also channelrhodopsin. So in this case, we have a transgenic mouse that is expressing channelrhodopsin, which is an excitatory opsin, under a specific promoter that is called M72, which is a promoter of a specific olfactory receptor, which is present only in specific neurons. And you can see here where those neurons are projecting into the olfactory bulbs. So on the other side, you can also use viruses. And how do you do that? Well, you can engineerize the DNA of the viruses to express the opsin of interest under a specific promoter. And then you can inject this virus in the area of interest. And both in the case of the viral expressing protein opsins, but also in the case of transgenic mice, what else do you need? Well, actually, to deliver light, you actually need an optic fiber that will allow you to target specific areas of the brain. So in the top of this slide, you actually have also representation of which kind of target in terms of spatial and temporal resolution you can have with optogenetics. And in particular, here you can see that the optogenetic allow us to target nuclei, but specific nuclei of the brain, but you can target also layers, specific layers of the brain, or neurons, and the different population of neurons. So this is the spatial target that you can have with optogenetics. And on the other side, you can also see the temporal resolution of optogenetics, which starts from a few, less than milliseconds to few minutes. And this depends on for how long you actually turning on the light to activate or inactivate your neurons, depending on the protocol, on the experimental protocol that you decide to use. So how do you target specific areas of the brain with viral injection? Well, you do need to have surgery on the animals, and we do have specific coordinates. Probably this part of the story you already know, but I didn't know the audience very well, so I decided to put also this slide. And eventually you can target specific areas of the brain just by, consulting a mouse brain stereotaxic coordinates and then placing the animal under stereotaxic, as you can see in one of those figures. And then you can target specifically, using those coordinates, the area of interest. And you can inject the virus specifically on the area of interest, and then in the same area you will place also an optic fiber to target the area in which the cells, by viral injection, will express your opsin now and can be controlled with light. So this is an example of one of the experiments that we did, and this is again using both a transgenic mouse and viral infection. So we had this crazy idea, and this is in 2019 when I was still a postdoc in Toronto, so in a laboratory in Toronto. Well, we had this crazy idea of trying to implant an artificial memory in mice. And so we thought that just by activating specific areas of the brain, we can actually mimic the an external experience. And in this case, our the memory that we tried to, and we actually succeeded in implanting, was an olfactory memory which was related to an aversive stimulus, so that we can test after the formation of this artificial memory just by placing the animal in front of the real odor if the animal was scared about it. And this is indeed what we were able to do just by using optogenetics. So we used transgenic mice in which we targeted this M72 glomerulus, which is a glomerulus that is activated, an olfactory receptor which is activated by the presentation of specific odors in mice. And at the same time, we targeted by injecting a virus in a region of interest and then placing an optrode on the right above the projections of this region of interest to another region of interest. I'm not going to go into the details of this, but this pathway from the lateral labellula to the VTA is a pathway that is known to be important for the expression of fear information in mice. And so the idea was that instead of like giving to them the real experience of the odor and of a fear of a shock, for example, we just activated those neurons into the brain by using optogenetic, by light, at the same time, simultaneously, so that if we could actually implant a fully artificial memory, these two information would be then associated together into the brain. And the next time that we presented the animal with the same odor, we would be able to see that the animal is fearing this odor, is avoiding this odor, which is indeed what we did, what we were able to do. And if you're curious about it, I can share with you the paper about it, and we can discuss it later. But indeed, this is for me just an example to show you that we can use both transgenic mice that are already expressing channelrhodopsin under a specific promoter, like in this case for the M72 olfactory receptor, but we can also inject a virus under the specific area of the brain, and we can target that specific area of the brain just by placing the optrode above that area, or even the projections that are going from region A to region B to target those projections. And here you can see a beautiful example of those projections. I was very, this is very fascinating to me. Neuroscience can be just fascinating just by looking into the brain and see what beautiful things you can find. So in this case, you can find it in pink, but in reality, the virus was expressing a yellow fluorescent protein. So we injected the virus in this area, that is the lateral labellula, and you can see these beautiful projections going from the lateral labellula to the ventral tegmental area. And then we targeted those projections just by placing the fiber above this area. So there are again, from 2010, that has been an explosion of different projects and different experiments that has been published using optogenetics. And this one is, to me, like one of very, very cool to see, actually. This is on Drosophila. And in this experiment, what they did was to try to inhibit the neuronal system that is involved in Drosophila locomotion. So they are inactivating a neuronal system using different type of inhibitory opsins, and that you can find on the bottom here, ACR1, ACR2, and the ENPHR. So what is their main purpose on this paper is to try to understand which opsin is better for inhibiting this system. And you can see that the normal activity of the animals, and then when the light goes on, you can see immediate stopping, inactivation of the locomotor activity in these animals, in both, and it's very, very clear in both the ACR1 and also 2. And eventually at one point, the light will go off, and you can see it by the color above. Yeah, here in this moment, the light went off, and you can see that the normal activity of the animals goes back as it's nothing happened before. So this is the cool aspect of optogenetic, is that we can tightly control the activity of the neurons, and we can tightly correlate it with the real-time behavior of the animals. This is another striking experiment that has been done in mice. And actually, they were controlling a specific area of the amygdala, which is important for controlling predatory hunting. So you can see normal behavior of the animal, the laser is off, and then when the laser goes on, you can see immediately the change of the behavior of the animal towards this piece of hood that basically, until then, it was totally unaffecting the behavior of the animal. And you can also see very clearly what is happening when the light is on. It's immediate hunting behavior, and then the light goes off and the animal is like, again, it's normal again. So we can study a lot of things with using optogenetics. And in my laboratory, for example, we are interested in head erection cells. What are these head erection cells? Where there are cells that are important in our brain. Even in humans, they have been discovered. And they are important for they are only responding to the head directionality of our head, the directionality of our head. So in this moment, my head is looking in this direction, and I will have some cells into my brain that are spiking, that are active, just because my head is in this direction. And if I move my head in a different direction, I will have another subset of cells that are responding now. And again, if I'm moving again, I will have another subset of neurons that are responding to this direction. And this is true also in mice. It has been found originally in mice. And this is such a fascinating type of cells that we are studying into our laboratory. And in this case, I just want to show you what we did with optogenetics. We tried to activate all those cells together, which was a crazy experiment because indeed you never had your head in all the 360 degrees direction. It's physically impossible. And so also the representation of our head erection system cannot be with all the cells active at the same time. So we did this experiment in which we kind of forced the nature of these cells to try to see what was the behavior of the animal. And here you have 3 examples, and you can actually, I spoiled you with the results here in the bottom of this video. But eventually you can see indeed that these animals are trying to cover with their head the 360 degrees when the light goes on. So it's very fascinating, but it's just an example of what we can really do with optogenetics to try to understand what is the activity of the neuron, how it correlates with the behavior of the animals. Okay, so I'm just gonna sum up the optogenetic information that I shared with you, and eventually, if I go too much into and then I move into the second topic, I hope it's not gonna take too much time, but you can stop me and we can discuss later on the different results. So yes, please.
I have a question. I read that multiple opsins can be expressed in the same cell. And I was wondering how we could take advantage of that to study behavior.
Yeah, so yeah, this is true. We can actually have multiple opsins, which can be, for example, excitatory and inhibitory at the same time within the same cells. And so by using different lights, you can see how the same cell is responding to activation versus inhibition. For example, what it has been done is to use those lasers in different days, or in different, even within the same day. You can turn on the blue light, see what is the response of excitation of that same subpopulation of cells to the behavior of the animal. And then the day after, or a few hours after, you just change the laser and you turn on the yellow light to see what is the effect of the same population of cells when you inhibit those population of cells in the behavior of the animals. Does it make sense?
Yes, thank you.
Sure. Okay, so I'm going to summarize the optogenetic stuff as I explained to you so far. So how optogenetic works is to actually use artificial viruses to add genes of specifically targeted neurons. The neurons will produce light-sensitive channel rhodopsins, which are channels expressed in the neuronal membranes. And shining light onto these neurons will make them fire, but we also saw that it's possible to both activate but also inhibit these neurons by using light. What is the strength of optogenetics? Well, both its temporal and spatial targeting of neurons to stimulate. So indeed, with lasers, we can target very small areas of the brain, and the specific neuron can be targeted using specific promoters. And this can reach the highest spatial precision that you can picture right now. And the second point is that it's temporal resolution, so light can turn on and off with the highest temporal precision, leading to a very fast kinetics of the activity of those cells that we can control with light. What are the limitations? Well, that it still requires transgenics or viral delivery, and so which means that you cannot just stick an optic fiber into the brain and you have the activity of the cells. You need to deliver a gene, a change in the gene expression within the cell of interest. But there are exciting new tools that are progressing. What is the main point about like this limit is the type of challenge that you can find when you want to change from virally infecting an animal model like mice to some specific models like non-human primates or humans, for example. But there are clinical trials for conditions such as retinitis pigmentosa, which is a condition of visual impairment. And there are some publications already that show that we can use optogenetics to modify the retina of some patients and restore indeed or improve visual deficits. So again, regarding the human neuroscience, primate and human brains are very large, which means that a larger area needs to be targeted, which is currently very challenging. Again, and also the other big limit is how ethically and can we perform optogenetic in humans in an ethical and also safe way. So I'm going to move into the second part of the talk, which is actually a technique that we are using, which is calcium imaging to visualize a neuronal activity. And this is a diagram very similar to the one that I showed you just for optogenetics that you can still find into this diagram about the spatial and the temporal resolution that you can find using different techniques. So you may have heard obviously about EEG, MEG, PET, and fMRI. So the interesting aspect of this diagram is to really immediately understand which kind of spatial resolution you can reach with this kind of techniques, which is generally very not specific at all. While with calcium imaging, that I hope everyone can find here into the diagram, it's on the bottom. So eventually, with calcium imaging, you can target nuclei, so big areas of the brain, layers, neurons, but also dendrites. And sometimes it is possible even to visualize spines just by using calcium imaging. And also the temporal resolution, it's very interesting because it allows us to visualize neuronal activity in terms of like at least less than milliseconds to 2 minutes, and sometimes even 2 hours. Hours is more difficult, but this is the kind of range that we can reach. So we can reach neuronal activity is not as precise as an electrode, the electrical field that you can study using electrophysiology, as we will see. But indeed, it allows other advantages related to these techniques that we are also going to explore. So how does it look like when you look at the calcium imaging? There are different types of calcium imagings. In our laboratory, we are using a miniaturized microscope that you can implant into the head of the animals. And here you can see a raw data, so it's not processed at all. You can see the behavior of the animal. The animal is freely moving into this task. And there is a tiny microscope on his head. And basically you can, while the animal is moving, so while the animal is normally behaving, you can actually see what is happening into a specific area of its brain, which is very fascinating. Trust me, when you see it in real life and you see the animal on one side and on the other one you have the screen, and you can really picture the activity of the cells, of all those cells. It's very, very fascinating to think that this is happening into their brain. I found it very fascinating in real time. So we are going back to the schema of the communication between cells and how action potential occurs. And the reason why I wanted to bring you back to the schema is what is the role of calcium when a neuron is activated. Well, it's such unique, like essential rule of the calcium, because without the entrance of the calcium, neurotransmitters cannot be released from the cells, so there will not be any communication between the cells. So calcium is a very strong indicator of neuronal activity, and it can be actually used as a proxy for electrical depolarization. So far, eventually, yes, you can still look at the action potential by just sticking an electrode into the area of interest. But you can also use calcium imaging as a proxy of this electrical depolarization. And how can you do it? By using calcium-sensitive proteins. So what are those calcium-sensitive proteins? There are different types of calcium-sensitive proteins. GCAMP is one of them, and is a calcium indicator. So this GCAMP is a genetically encoded calcium indicator, and is also called GECI. So the GCAMP is consisting on different pieces, and in particular we have a green fluorescent protein, which is the classical GFP protein that you can get from jellyfish. And there are also other components like the calmodulin component that you can find here, which is a component that is essential because it allows the connection with the calcium. So when the calcium is present into the cell, calmodulin will connect with the calcium. And then there is also the calcium interacting, the calmodulin, sorry, interacting M13 peptide. So how does it work, this system? Is that when calcium is present, calmodulin undergoes a conformational change. And the region that is now present due to the this change in conformation will be able to hold specific proteins, like, for example, the M13, which is a very special protein because it's preventing water molecules from accessing this chromophore, the GFP. So why this is important? Well, because in the absence of calcium, the circulating perturbated fluorescent protein exists in a very poor fluorescent state. So we cannot indeed see it. And here in the graph on the right side, on the bottom, you can see what is happening when you turn on the light. So you shine this area with the blue light, and you should have the response of the GFP. But without calcium, this response of the GFP is very low, and this is because of the conformation of the GFP, which is in a poorly fluorescent state due to a water pathway that enables protonation of the chromophore. And so there will be a poor absorbance of the excitation wavelength. However, when calcium is binding to the calmodulin, this results to the structural shift that eliminates the solvent pathway. And so that will be a brighter fluorescence when in presence of calcium. So what is the difference again between calcium imaging and electrophysiology? There are some studies that have tried to combine these two techniques, so electrophysiology and calcium. And as you can see, actually, in gray, and this is on the top of this slide. In gray, you can see the change of action potential of one single cell, and you can see how many action potential you have every time. And on the top, you have in the black line, the calcium imaging. So the fluorescence, the change in fluorescence, and you can clearly see that there is a difference between the two. But indeed, just by computing those information, you can clearly understand how much of action potential every single cell had. So again, this is not a clear response as you can have in the electrical field when you just stick an electrode into the brain. But what you can see is, what you can have is a proxy of neuronal activity. And here you have two examples, which are GCAMP6S and GCAMP6F. And this depends on the kinetics of fluorescent intensity changes. So you can have different type, and here you have in B, in this graph in B, you can have different type of GCAMP, so of calcium indicators, and you can have different kinetics of calcium indicator. And depending on the type of experiment that you are interested in, you may consider to use different calcium indicators. Also, on the bottom left of this slide, you can also find another graph. And here is interesting to notice what is the kinetics of calcium. So how calcium enter into the cell and then exit into the cell. And these kinetics are way faster than what we can get from calcium indicator. But things are going better and better basically every year. So you can see two examples in green and in blue. And in green, the 6S, it's a an older calcium indicator that we all laboratory used to use. And now there are newer calcium indicators like the 8F, which we are using right now, which have better dynamics. So with time, probably we will have even better calcium indicators that can allow us to really visualize calcium influx in real time. So the different type of calcium indicators, and here is just a sum up of the different aspects, the differences that we can find in calcium indicators are, for example, the sensitivity of binding calcium, the excitation emission spectra, the brightness of those indicators, and also the kinetics, as we discussed, of fluorescent intensity changes. So here there is just an example that I took from Miscopics, which is a company that is selling the miniaturized microscope. So here is a brain, a representation of a brain, and in blue you can find the cells that basically we want to target in this specific experiment, potential experiment. We inject the virus, GCAMP is now expressed into these cells. We can place a lens right above this area, and then a base plate that we can then attach our microscope, and then we can turn on the laser to then have the response of the fluorescent indicator, that is in our case GCAMP. So there are different components of this microscope. This is in all ways a miniaturized microscope. So there are different components that are important for the imaging, for the LED, to activate to visualize the fluorescent indicator. There are optical components, and then also we have a green lens that we can use actually to be attached to the main lens of the microscope, that we can use by placing this green lens in even very deep areas of the brain, so that we can really target potentially with the miniaturized microscope every single area of our brain. We don't need to remain superficial into the brain, so we don't have only the possibility to look at the cortical areas, but we can again go very deep into the brain and really target every area of the brain. The cool thing about the miniaturized microscope and the neuroscience in general today is that a lot of those tools are open sources. So you can even build your own miniaturized microscope. You can build just the different, you can build it by buying the different components of the miniaturized microscope, and you can eventually even customize it to make it better for the performances that you are looking for your specific experiment. And another cool thing about it is that since this is an open source technique, there is a huge community of people that are using it throughout the entire world. And there is a lot of communication between this community to try to improve, to find problems about these techniques and how to make it better and better as much as we use it. So the other components of the miniaturized microscope is obviously the possibility to connect it with the behavior of the animal, which is the main point. We do want to visualize the neuronal activity while we are looking at the behavior of the animals. And so this is the reason why we want to connect the head-mounted scope with the DAQ archway that is connecting also, that is connected with the computer. And on the other side, at the same time, we have the visualization of the behavioral behavior of the animal through a simple behavioral camera. Here is an example from our laboratory, actually, of a mouse freely moving. The activity of those cells. Then, when we can process the imaging that we get from those studies, we can process them. We can delete the background. We can delete the movement of the camera, because obviously this camera is moving together with the behavior of the animal. And then in real time, we can actually track the changes in fluorescence, and we can raster plots of the calcium events that occur for every single cell that is present, is present into the field of view. So there are several things that we can do with it. This is just one example from my laboratory, from two talented postdocs in my laboratory, which are Jose, which left this year, actually, the laboratory, and Clément. And what they did was to try to understand how neuronal activity correlates with a fear memory. So we train animals in different paradigms. For example, in this case, the animal is just freely moving into an open field. And here, close to the video, you can see just the head directionality of the head of the animal. And you can correlate the head directionality of the behavior with the activity of the cells, so that we can then compute those information and try to find if there are head direction cells, so the one that I just explained to you, to find the head direction cells within the cells that we can detect using calcium imaging. On the other side, you can correlate the activity of the cells with a lot of other things that happen to the animal. For example, if we shock the animal in a fear paradigm, we can see that there are some cells that are strongly responding to the delivery of the shock. And there are a lot of other studies that has been done using a miniaturized microscope. For example, it has been used, the calcium imaging, so the miniaturized microscope, to identify place cells, which are very similar to the head direction cells, but instead of responding to the head directionality, they are responding to the position in which I am. So in this moment, I will have some place cells that are responding to my position in the space. But if I'm moving in some other direction into my room, I will have other cells, other place cells that are responding to this position. And it has been shown that we can actually identify those place cells using calcium imaging. What is the cool aspect of calcium imaging that we cannot reach with electrophysiology is that we can track those cells over time. And I think I do have a slide about it, which I'm going to go through right now, and then I'm going to go back. And so what we can do is to actually, since we have the visualization of those cells in the space, in our field of view, which is corresponding to the lens area of the microscope. So what we can do is to visualize those cells, and then the day after, or even 30 days after, we can go back, visualize the field of view, and find the same cells that were active 30 days after, 30 days before, and see how those cells are now responding to the same behavior, or to a different behavior, or to a memory of that behavior. So that's the reason why, for example, using calcium imaging, we can detect place cells, and we can, for example, detect even head erection cells, which is something else that we are doing in our laboratory. And we can detect if those place cells remain place cells the day after, or 30 days after, and if they do still respond to the same place field that they were responding days before. We can also detect other things using calcium imaging. Again, we can detect if a cell is responding to a shock, but we can also detect, for example, if the animal is moving through a specific task, if there are some cells that are more active when the animal is in the open arm or in the closed arm. And this is why this is relevant. Well, for example, because we are studying if the animal is anxious, if it's stressed, and when the animal goes into the open arm, it means that the animal is more willing to explore, so it's less stressed. And we can make some correlates about the behavioral state of the animal itself. So this is another experiment that I did that we are doing in our laboratories, and a cool aspect of this experiment is that this is just another way to represent calcium imaging. So every line here represents a specific neuronal activity, and we can detect cells that are preferentially active in specific moments of the activity of a specific task. So, for example, cells that are more active during the shock delivery, cells that are more active before the shock delivery, when the animal is in freezing behavior, when the animal is not in freezing behavior. And then we can track those cells at different time points, and we can see how those different cells are responding to a memory. And also, this is one aspect of the calcium imaging which is unique, which is the possibility to visualize the same neurons over time. But another very interesting aspect of calcium imaging is that since we can visualize where the cells are, we can then study the topographical relationship between neurons. I'm going to skip this part because it's a bit too complicated, but we can go back later if we have, if you have any question. And I just want to conclude with the key points to understand the optical methods, which is that the electrical activity of neurons can be imaged using fluorescent molecules, that calcium-sensitive indicators fluoresce more in presence of calcium, which correlates with the neuronal activity increase, that imaging neuronal activity makes possible to see more cells simultaneously with large field of view, to target specific cell types by genetically encoded indicator expression, to track the same cell across time, to visualize the topographical relationship between neurons, and to measure neuronal activity with less bias towards more active cells. So what is specifically this point, I didn't have time to explain to you, but eventually with electrophysiology, because we are doing spike sorting, some of the cells that are really not active at all or very slowly active, maybe they can have a spike every minute or so, it's probably they are going to be not considered into the analysis if they have very less activity. But with calcium imaging, since we are visualizing all those cells, we have less bias towards those cells. And finally, the optogenetic is another approach that I explained to you in the first part of the class. And this is using light-sensitive ion channels to manipulate, which can be both activation or inhibition of cells directly by light. And with this one, I will conclude. I will thank you for your attention.